The overall goal of the following experiment is to develop a next-generation photosensitizer for photodynamic therapy, or PDT, application in cancer therapy. This is achieved by adapting a reprecipitation method of fabrication to make water-dispersible conducting polymer nanoparticles. As a second step, the nanoparticles are incubated with cancer cells in vitro, followed by treatment with light to generate reactive oxygen species, or ROS, which are the active radicals that partake in PDT. Next, treatment effectiveness is evaluated by quantitative assays and qualitative imaging to determine live versus dead fractions of the cell population. The results show that aggressive cancers, such as ovarian cancer, modeled here by OVCAR3, abundantly take up the nanoparticle photosensitizer reported in this work, and that the PDT treatment is highly effective in vitro in that case. The PDT effectiveness correlates directly with the amount of nanoparticles taken up by the different cell lines.
The main advantage of this technique over the existing methods like chemotherapy, radiotherapy, and surgery is that the treatment can be target-specific towards disease site to reduce the side effects. The active material used shows no in vitro cytotoxicity, and further development will make the nanomaterial system more specific towards the disease site.
Begin this procedure with culturing of cell lines and preparation of the MEH-PPV stock solution as described in the text protocol. Add 50 microliters of the undiluted MEH-PPV stock solution into 3 milliliters of tetrahydrofuran, or THF. Label this